Hi everyone. In this fourth video on dissolution science series, we will discuss choosing right dissolution medium, pH, surfactants and buffers. The dissolution medium controls how much drug dissolves and how fast it does. A wrong medium can make the method non-discriminatory or clinically irrelevant. That's why medium selection directly impacts solubility, reproducibility, and clinical predictability. Now, key criteria for selecting medium. The dissolution medium should match the API solubility profile and pK to ensure proper solubilization and sink conditions. It must also reflect dosage form, behavior, whether IR, ER, floating, or poorly soluble, and remain physiologically relevant, such as gastric or intestinal pH. Most importantly, the medium must be discriminatory and regulatory compliant, meeting USP and FDA expectations. Drug solubility is highly pH dependent, especially for ionizable APIs. Weak acids dissolve better at higher pH, where they are ionized, while weak bases dissolve better at low pH, such as in gastric conditions. The drug's pK determines how much is ionized at a given pH, making the pH-pK relationship critical for predicting solubility and selecting the right dissolution medium. Always link pH selection to the drug's pK. For example, weakly basic drugs may dissolve well in the stomach but precipitate in the intestine, which is why multi-stage media are often justified. Common dissolution media aligned with USP and FDA guidance include pH 1.2 gastric simulation, pH 4.5 intermediate transition, pH 6.8 intestinal simulation, pH 7 to 7.5 neutral or colonic conditions. For drugs with pH dependent behavior, a multi-stage approach is often used. pH 1.2 to 4.5 to 6.8. Buffers control pH stability during dissolution. Always evaluate buffer capacity, especially for high drug loading where stronger buffers may be needed to maintain target pH. Surfactants should be used only when truly needed. They are appropriate when physiologically justified or required to maintain sink conditions for poorly soluble drugs. Avoid excessive surfactants levels as they can artificially increase dissolution rates and mask the real formulation differences. When selecting surfactant concentration, always start with screening study. Begin low, typically 0.1 to 0.25%, and increase gradually only if sink conditions are not achieved. Avoid going far above the critical Michelin concentration, as this can mask formulation differences. The final concentration must be scientifically justified using solubility enhancement and discrimination data. It's also good practice to check the FDA dissolution database for reference product benchmark. Dissolution volume selection is critical for maintaining sink conditions. Typical volume are 500 ml, 900 ml or 1000 ml. A sink condition means the volume is at least 3 times the drug's solubility. If sink conditions cannot be achieved, add a justified surfactant. Use USP4 flow-through cell apparatus or increase volume if scientifically justified. Maintaining sink condition prevents medium saturation and keeps the dissolution rate reliable and measurable. Biorelevant media are used as an optional advanced stage, especially for complex formulations and modified release systems, or when recommended by FDA. Common examples include FASSGF, fasted stage gastric fluid, FASSIF, and FESSIF, fasted stage and fed state intestinal fluid. Benefits include improved IBIBC and deeper mechanistic insight. Challenge are highly complexity and cost. When selecting a dissolution medium, bring everything together. API solubility and pK, target site of drug release, ability to maintain sink conditions, discrimination between formulations, aligned with regulatory expectations. A good medium is scientifically justified, clinically relevant, and regulatory ready. Regulatory agencies expect biorelevance, discrimination, and scientific justification, not arbitrary media choices. Guidance from the USP, including USP 711, USP 1092, and FDA product specific guidance, clearly reflect this. Any use of surfactants must be justified, and the method should demonstrate discrimination and stability relevance. Now to summarize, selecting the right dissolution medium is a balance of science, physiology and regulatory expectations. pH is chosen based on API solubility and GI relevance. Buffers are used to stabilize pH. Surfactants are added only when necessary. Volume must support sink conditions. Above all, every choice must be scientifically justified. Thank you for watching. Please subscribe us for getting update on such industry application oriented videos, and stay tuned because in this 62 video series we will dive into dissolution method development, validation, troubleshooting and advanced approaches, real industry case studies. In the next video, we will cover sink conditions explained, how to achieve and maintain them. If you have any question, please post them in comment section. I will do my best to respond. See you next video. Thanks.